Great, thank you, and thank you for the invitation to come speak today. This will be a bit of a fish out of water compared to everything else that's gone on today so far. This is all semiconductor nanocrystals, absolutely nothing to do with plasmonics today, although I promise a little bit more of that in my talk tomorrow. So my focus here is on semiconductor nanocrystals, otherwise known as quantum dots. And for decades now, researchers have used quantum size effects to control the properties of quantum dots. The focus of my talk is how we're evolving as a field towards doing things that are a bit more interesting, perhaps more complicated than that. We still utilize the quantum size effects to create properties, but on top of that, we're employing nanoscale engineering, control of structure at the nanoscale inside the quantum dots to facilitate emergence of new properties, the addition of new properties for multifunctionality, etc. And so I hope to give you a flavor of where this field is going in this talk. And we can also title this talk slightly differently, Can Old Dogs Learn New Tricks? Okay, so everybody knows you can control the size of a quantum dot and change the semiconductor band gap, but we want to do more than that. We want to teach these things to be even more interesting than they already are. And so I want to highlight some of the important or key collaborators in this work. I'm a synthetic chemist, so we synthesize these materials, but without the aid of spectroscopists, physicists like Han Toon and Anton Malko, we wouldn't know nearly as much as we do about them. We also work with theorists like Andrei Pirutinsky from Los Alamos, and also more recently, electron microscopists like James McBride at Vanderbilt University. So as a brief outline to this talk, I'm going to describe functional nanocrystal quantum dots, and specifically the flavor that we invented effectively a number of years ago now, the giant quantum dot, or GQD. These were engineered at the nanoscale to be fully non-blinking now. And I'll briefly describe how these came about and how we've moved beyond visible emitting giant quantum dots to the near infrared, which has some more relevance to bioapplications. Theory, how we use theory to guide our discovery and development of new giant quantum dots, and how we're beginning to even move beyond the spherical nanostructure to more complicated shapes such as asymmetric tetrapods, and how we can still use the same general mechanism underlying the non-blinking behavior in giant quantum dots to achieve similar functionality in these unique structures. And then finally, I'm going to end with a need for an example of new tools that we're using to advance the functional nanomaterials discovery, and specifically in the area of correlated optical, structural, and compositional analyses. And so this is a QD story, as I've already warned you. I hope you will see that the quantum dot story is highly complementary and has synergy with plasmonics and also with bioapplications. And so tomorrow, I'll show how these very functional and stable quantum dots can be used in hybrid plasmonic quantum dot nanostructures to create new multifunctionality, and also how the ultra-stability of the giant quantum dots can be used as a molecular probe in applications where you want to track a molecular species, for example, in living cells over long periods of time. Obviously, the non-blinking and non-photobleaching behavior that I'll show you of these new types of quantum dots aid in those applications. So very briefly, since I think this is a plasmonics community here, what are quantum dots? I've already alluded to this. They're basically zero-dimensional semiconductors where the excitons or electron hole pair that's excited or created by photoactivation is confined in three-dimensional space. This quantum confinement leads to our ability to tune the electronic properties of the semiconductor, namely its band gap, and by simply changing the size of the particle in the quantum confinement regime, we can tune the band gap in the case of cadmium selenide nanocrystals all the way from the blue to the near infrared. In the case of an infrared band gap bulk material like lead selenide, we can actually tune from about the near infrared of a micron to even 4 microns, so all the way fully into the mid infrared. So the same composition, change size, change semiconductor, change color. And so what can we achieve? We achieve fluorescence that's very efficient typically, that's narrow band and bright. Absorption is broadband, and so these particles have a large absorption cross-section and they're easy to excite. And significantly, for the purpose of applications, all of these are produced at low cost and in scalable solution phase syntheses. And we like to refer to these as high-tech materials from low-tech methodologies. The resulting particle size distribution and therefore the resulting quality of the photoluminescence obtained from them is easy to control in a very monodisperse fashion where we can achieve particle sizes that are distributed by plus or minus 4 or 5%. And the particles are single crystalline and made solvent suspendable by the application of a ligand monolayer on their surfaces. So not unlike many of the gold nanoparticles that you're very familiar with. So I want to highlight now a property of standard quantum dots that I think was already mentioned by one of the earlier speakers. That is blinking and photobleaching. So conventional quantum dots, if you add a wide bandgap semiconductor shell onto the outside of the core emitting nanocrystal, in this case, let's say it's a cadmium selenide core and a zinc sulfide shell, you can drastically improve the quality of the emission that you achieve from the cad selenide core. The quantum yields can approach 80 to 100%, so very, very bright. Yet, these nanocrystals, even though they exhibit this nice feature of bright photoluminescence, still have the annoying feature of blinking. And so what I show here on the right is actually a video of standard core shell quantum dots blinking. You can see that the individual specks of light actually represent individual quantum dots that are turning on and off in a random fashion. This fluorescence intermittency is also shown here in this yellow box. This is a intensity time trace for a single quantum dot turning on and off, i.e. blinking, but it's also photobleaching. So over a certain amount of time, the wonderful qualities that you like in the quantum dot that you've purchased, maybe from life sciences, will disappear. And if you're using these as a molecular probe, of course, your probe is disappearing on you as well, and that really limits your tracking times, for example. And so what we've done is to develop a type of quantum dot that doesn't blink and it doesn't photobleach. And we called these quantum dots back in 2008 the giant quantum dot. And giant, not meaning that they're that terribly large. They're still about 15 nanometers in diameter, but they are bigger compared to their standard core shell counterparts. And the example I show here is a cad selenide core again, but now the shell is not zinc sulfide, it's cad sulfide, and it's also not thin, but very, very thick. And what you see over here on the right is a movie. It actually is running, and it runs forever because these things just don't die. But the particles are not blinking on and off. At most, they're twinkling slightly because their intensity is fluctuating, but we're not actually losing photoluminescence entirely. And so this is a single quantum dot time trace of intensity versus time, and you can see the non-blinking behavior as well as the non-photobleaching behavior. And so fundamentally, a new type of quantum dot with respect to function that I would argue has important ramifications for biology. But before I get to that, I just want to briefly say something about the mechanism responsible for blinking, because if you understand the mechanism for blinking, you can think about synthetic strategies for shutting it off. Okay, so the conventional wisdom for fluorescence intermittency, where you see the intensity turning on and off in a random fashion, has for a number of years been the following. That quantum dots randomly cycle through uncharged and charged states, that they're literally becoming either charged with an excess electron or an excess hole. And when they're charged, the quantum dot will be off or blink off due to a very efficient non-radiative process known as Auger recombination, which I show schematically over here on the right. If you look at the left diagram, you see the case for the quantum dot that has an extra electron in it. And so the energy of recombination of the electron hole here is not given off as a photon, which is what you'd like, but instead gets transferred to the extra carrier, the electron here, which jumps up in energy and then dissipates the excess energy in the form of heat. And so this whole process is a dark process and occurs when the quantum dot is charged. And so that, for a number of years, was viewed as the mechanism underlying blinking. And we actually believe that that is still a principal mechanism responsible for blinking. But there's actually an additional process that we discovered a few years ago now, as shown in this paper here, where we call the conventional method type A, and there's a new method, or there's a different additional process called type B blinking. And we discovered this by developing a technique of spectroelectrochemistry that allows us to look at a single quantum dot and its optical emission under photoexcitation, but also in the condition where we can actually selectively charge and discharge the quantum dot electrochemically. And so when we do this, we can very precisely understand what is happening to an individual quantum dot with respect to charging, and how that correlates or does not correlate with the blinking process. And so as we expected, we saw the typical A-type blinking behavior where, as you see here in this plot, the average lifetime is actually correlated and fluctuating with the intensity of this quantum dot over time. And so what that means is when the intensity is down, i.e. the particle is blinked off or close to blink off. These were quantum dots were constructed so that they didn't entirely go dark. They go to a gray state or a dim state. So when they're in the dim state, their average lifetime is short, and when they're bright, their average lifetime is long. And there's an explicit correlation between the darker states and a faster lifetime, and a brighter state and a slower lifetime. And so we believe this is it really can be attributed to the Auger-mediated blinking process, where one would expect that if Auger is active, then your lifetime would correlate with the intensity. In contrast, we saw very different behavior over here in the case of this quantum dot, where the intensity is fluctuating, but the lifetimes are not. And so here we needed to invoke a different mechanism that didn't involve charging and didn't involve Auger recombination. And what we believe is happening here is that hot carriers, and this is what's shown here as a hot electron, a highly energetic electron, is just jumping out into a surface state temporarily and then coming back in. So basically we're circumventing a luminescent process, but we're not impacting the lifetime of the process because Auger is not active here. And so these two processes, we believe, are fundamentally underpinning the process of blinking. And so so I'm going to look at that in a second. And why is blinking important? In the area of biology, for bioimaging, biotracking, blinking and charging related bleaching, of course, is a challenge, especially if you're interested in using the particles as molecular probes. We're also at the lab interested in single photon light sources. Blinking obviously defeats your ability to obtain a photon on demand when you want it, because you never know when the particle will be blinked off if blinking is active. Lasing, if Auger recombination is efficient, will defeat your need to build up bioexcitons to achieve the regime of gain. So lasing is shut off. And then finally, solid state lighting. Any non-radiative process really defeats your ability to achieve high efficiency. So for all these reasons, we want to address these three issues of blinking, charging, and non-radiative Auger recombination. Okay, so now we know that blinking, that there are two mechanisms responsible for blinking. One relates to charging and Auger recombination, and the other relates to carrier trapping to surface states. And so synthetically, one needs to come up with a plan for how to deal with all of those characteristics, all of those processes through nanostructuring. And so we look first to the issue of Auger-mediated blinking processes, and we think, how can one suppress Auger? We already know from prior experience that large volumes reduces exciton-exciton coupling. That impacts Auger. Type 2 band alignments, and so if you have reduced electron hole overlap in your core shell nanostructure, as shown here in these two right examples, the carriers are separated in space. One is in the shell, and one is in the core. Then you reduce repulsive coulombic interactions, and also to help to suppress Auger. And finally, theory by Sasha Efros a number of years ago showed that if you could smooth the confinement potential at the core shell interface, you could potentially also reduce Auger. So all of these are knobs that we can use to tune the structure and thereby tune the Auger-mediated blinking process. And then second, the other one's conceptually simpler. If you just want to reduce carrier trapping to the surface, grow a thick shell. Physically separate your core, where the electron hole carriers are, from the shell surface. And so by combining all of those characteristics, we came up with the structure shown here on the bottom right, which is the core thick shell, and specifically in a quasi-type 2 and type 2 band alignment format, where in this case the hole is confined to the CAD selenide core, and the electron is loose and free to move about the CAD sulfide shell. Our thick shell is reminiscent, we think, of an epitaxial quantum dot, where these physically grown nanocrystals have been known to show strongly suppressed Auger recombination and also blinking. And so we're effectively in solution creating a solution phase epitaxial quantum dot. And we do so utilizing a synthetic method originally developed by Xiaogong Peng called SILAR, or successive ionic layer absorption reaction. This method allows one to add monolayer by monolayer in a very refined fashion your shell material to the nanocrystal core, and to do so in an epitaxial fashion, and to create very large structures, at least from the nano's perspective, that are fully single crystalline and largely defect free, which is critical to avoid other non-radiative processes due to trapping. So we've developed this method originally in 2008, but since then have optimized the process by tweaking various reaction parameters, as shown here. If you're interested in the gory details of the synthesis, I would suggest looking at this Jack's paper. But the important thing is, from a functional standpoint, is that we went from a synthesis that gave us about 30% non-blinking quantum dots to 100% non-blinking quantum dots, or very close to that. And so now we know the recipe, so to speak, for achieving quantum dots that are really genuinely fully non-blinking and also non-photobleaching. And so what I'm showing here, I should point out, because you'll see a couple of these later, these histograms are the way we represent the full population of a giant quantum dot sample. And so this is non-blinking fraction and percent on time. And so the larger the bar on the right, where you're close to 100% on time, suggests that more of your population is non-blinking. And I want to point out, with respect to sort of nanostructure engineering, it wasn't just tweaking, precursor choice or ligand dynamics and ligand concentration that allowed us to achieve the optimized giant quantum dot, but actually we found that there was a significant role to the total particle size, so the volume effect. As I show here, we found a volume threshold. So depending, if you started with a small core, you'd have to grow to a thicker shell to meet this volume threshold of non-blinking behavior, or you start with a larger core and you ultimately need a thinner shell. In either case, at about 750 cubic nanometers, our non-blinking behavior turned on and then took off and then increased as a function of the total particle volume. But again, there's contributions from both the core size and the shell thickness. And then a follow-on study with collaborator Han Toon allowed us to extend our investigation beyond just blinking behavior to something potentially more interesting, and that of biexciton quantum yields. And these are a direct reflection of how well we've suppressed Auger recombination. And what we found here is that indeed the large volume particles allowed us to suppress Auger recombination, as indicated by achieving high biexciton quantum yield. So if two excitons can live in a single nanostructure, that means you've suppressed Auger. If Auger is active, those two biexcitons will effectively annihilate one another in non-radiative recombination processes, similar to the example of the extra charge case. And so if we can have biexcitons living for very long times in the quantum dot, then that means we've achieved Auger suppression suppression. And what we found here is, interestingly, there wasn't just a volume effect. What we found was there was a very strong correlation with the size of the core of the quantum dot and the high bioexciton quantum yields. So for this, actually the same volume, so a small core and a thick shell, or a large core and a thin shell, we actually achieve very high bioexciton quantum yields in the case of the larger core. And we think that underlying this is probably a mechanism related to how the core size affects the hole confinement in these type 2 or quasi-type 2 electronic structures. But anyway, again, there's a lot more to say than what I can show here in this short talk, but I would show you or point you to those papers if you want to understand more about Auger processes in the giant quantum dots. Okay, so now, for bio applications, it's likely important to move beyond visibly emitting CAD selenide, CAD sulfide giant quantum dot structures that give us nice red emission. But if you really want to push into the near infrared, you need to look at different compositions. And so we started investigating some indium phosphide nanocrystals and found that if we follow the hypothesis that a thick shell paired with a type 2 electronic structure should help us to suppress blinking, both through Auger-mediated processes and through the type B blinking processes, then we might actually be able to develop a sort of a larger family of giant quantum dots and that type of non-blinking functionality. And so here we look at indium phosphide paired with a CAD sulfide shell. This is calculated to be a fully type 2 infrared emitting system where the electron is fully confined to the core and the hole is confined in the core and the electron is in the CAD sulfide shell. Recombination across that interface releases an infrared photon. And what we find is if we grow the shell sufficiently thick, the emission intensity, again, like in the CAD selenide, CAD sulfide case, is non-blinking and is also significantly more photostable than before. This, to the best of our knowledge, was the first example of an infrared emitter that was looked at with a standard detection system at the single dot level. And so, which historically has been quite a challenge to take an infrared emitting quantum dot and actually see it at the single particle level, blinking or not. And so we saw, obviously not in this case. We also calculated the bioexciton lifetimes and again saw a correlation with the shell thickness, although the efficiencies in this case were not as high as in the CAD selenide, CAD sulfide system, so our Auger is not particularly suppressed. And yet, we get blinking suppression. So in follow-on studies, we determined that the indium phosphide CAD sulfide system, when it blinks, is dominated by type B blinking behavior, so not Auger-mediated blinking. So that's an interesting convolution of characteristics where we have suppressed blinking, but we haven't suppressed Auger. And so bioexciton lifetimes aren't particularly long. And so what we can achieve in this particular system is a near ideal particle for, say, single photon source applications. And I demonstrate that with this bottom right G2 figure. This is basically the distribution of photons at a detector over time. And when bioexciton efficiency is high, this middle or time 0 point will be very high as well. So that would mean that you're getting excitons and bioexcitons hitting your detector simultaneously. And for single photon source applications, that's obviously not what you want. And so what we have here is fully anti-bunch behavior paired with non-blinking behavior. And so almost an ideal single photon source. And so that development was guided by our basic hypothesis about the nanostructure that one needs to achieve suppressed blinking. And so we're kind of continuing along those lines, but now we're using theory to more explicitly guide our selection of core composition, shell composition, core size, and shell thickness. And so I don't have time to go through all the details, but this sort of theoretical foundation was developed for us by Andrei Pirutinsky and then we targeted these two systems, cad selenide, zinc selenide, and zinc selenide cad sulfide, to achieve suppressed blinking behavior in new systems based on a quasi-type 2 system, and then also another type 2, or fully type 2 system. Unfortunately, synthetically, these were much harder to achieve or to realize than our previous systems had been. The cad selenide, zinc selenide system is complicated by a tendency for alloying. So if those of you who have ever worked in these heterostructured nanomaterial field, you'll know that sometimes the chemistry you get out is not what you're trying to put in. And in this case, we got a lot of mixing between the metals, or the cations, and so we ultimately found the synthetic conditions to achieve the core shell structure, but they were not quite ideal. And then the next system was quite intriguing. It was complicated by a tendency of the cadmium to replace zinc. So there's a cation exchange issue here rather than alloying. But regardless, one can always find some synthetic pathway to achieve the structure you want. And ultimately, we did so by modifying the reaction conditions, in this case by reducing temperature and utilizing a single source precursor to avoid the cation exchange processes. So two new giant quantum dot systems, both with at least some degree of suppressed blinking behavior. And then finally, with respect to the examples of nanostructures used to achieve suppressed blinking behavior, I want to show another one that's based on cad selenide, cad sulfide. But in this case, we're moving away from the simple spherical constructs of a core shell nanomaterial. And here we have a core, but now the core has arms. And so these structures are called tetrapods. There's a cad selenide core with 4 cad sulfide arms, appendages from it. But the interesting thing here is that if we compare different lengths of arms and different thicknesses of arms, we see suppressed blinking behavior only in the case of the very thick arm structure. And interestingly, this thick arm was created not by traditional tetrapod synthetic methods, but actually by thickening the arm through Sylar. So very much like the cad selenide, cad sulfide giant dot case, we have a sort of a new giant tetrapod that is now showing significantly suppressed blinking behavior, but as a strict response to the geometry that we've implemented. Okay, and then so another interesting thing about tetrapods, so why move away from core shells and move into something kind of exotic like the tetrapod? It's known for a few years that the tetrapods, because of this extended arm length, can actually physically segregate excitons that are formed, electron hole pairs that are formed in the shell or the arms versus the core. And if you basically if you fill the states in the core, then you give a chance for the excitons that are sitting in the arms to actually to emit. And so you can actually get 2 different colors from the same nanostructure. You can get a red emission from the core and now a green emission from the arm. And so we explored that for the first time at the level of a single tetrapod. And what I show here is the onset of the second emission or second color, the green color of emission, as a function of pump power. So as you increase the excitation power of your laser source, you state fill the core, and so it can't accommodate anymore, but the arms are happily there to accept more excitons, and at some point they're able to photoemit, and you can produce 2 different colors from the same structure. And the interesting thing is, again, as a function of arm thickness, we can actually see non-blinking behavior from both the arm emission, the green emission, and from the core, the red emission. And so, okay, yeah. One more minute, and I just want to make a comment that the only way to discover and develop these new functional nanostructures due to their complexity is to really have an explicit understanding of the correlation between the structure and the functionality. And so far, that has been done mostly by guessing. And so now we're working with a group at Vanderbilt and a new postdoc to really push a new technique that was developed originally by the group at Vanderbilt. And this is using the fluorescent polystyrene beads as markers to tell you where on a TEM grid the nanocrystals are, so you can do correlated optical microscopy with the TEM. And what we're able to do in our laboratories is to really advance this significantly, so that on a single structure where you have the fine atomic structure, the elemental composition, you can really dig into the optical properties from blinking to G2 to lifetime, etc. And so that's it. Okay, thank you.